So, welcome to the last of our lectures for this semester. And you started out this semester in your class learning what organic molecules were, the difference between between the covalent bonds and ionic bonds and hydrogen bonds. And a semester later, into the semester, you're learning the intricacies of metabolism. So you should be proud of yourself. It's quite an accomplishment in one semester. But for this last slide set, we're going to cover three main things. We're going to talk about how we degrade fatty acids, right? So from triacylglycerides, perhaps, all the way down to the individual fatty acids, and then those turn into acetyl-CoA. And the next smaller part, we'll talk about what you can build from that acetyl-CoA, such as ketone bodies. And then the last part, we'll take some acetyl-CoA and see how it's stored as fatty acids, right? We'll build a fatty acid. Okay, so let's start with the beginning and see how we degrade the fatty acids. So first of all, what is a fatty acid? How is it stored? It's stored as a triacylglycerol, sometimes called triacylglyceride. And we talked about these before when we did the lipids lecture. And it was a glycerol molecule, as you see on the right there, the three black colored carbons. And it has three green fatty acids attached to it, on carbons 1, 2, and 3. And if you remember, those are three ester bonds. Okay, so where do we store this in the body? Right? Triacylglyceride is an excellent storage form of fats. So where do we store it? Well, mainly in two places. We store it as subcutaneous fat under the skin, and in visceral fat, which surrounds organs, internal organs. So what is the purpose, other than storing molecules for me? What is the secondary purpose of that subcutaneous fat, the fat found under the skin? So what do you think? Why store it there? Say that again? If you're trying, we can't hear you. So why do we store fat under the skin? What's the other reason to put it there?
Warmth.
Right, so it serves as a heat insulation, right? So it protects you or protects the body from losing too much heat, right? So we can stay warm. That's exactly right. So what's the point of storing it around all those internal organs then? It's clearly not to conserve heat, because you're not going to lose much heat from one organ to another inside the body. So why store fat around those organs?
Protection.
Protection from what? It's not from getting cold, like the subcutaneous fat is. So what are you protecting from? You're right, but what's the protection for? So you have this layer of fat around these internal organs that's going to cushion them from any stress, so a vibration or injury from a collision. So if it didn't have that, and you were tossed around the room, or you ran into a wall, get in a car accident, or you fell from a tree, or whatever it was, some kind of shock to the body, physical shock, those organs might bounce off of other things, right, or tear. And one of them isn't so well protected. So for your future medical professionals, when you have this person come into the hospital, or you come across someone on scene in an ambulance and they've been in a car wreck and it's clear they're alive, what organ is most likely to be damaged in a car wreck? Internal organ. It loves to explode. Anybody have an idea? It's because it doesn't have a lot of this protective fat layer around it. The skin of it's very thin. Go ahead.
Is it the spleen?
The spleen, yes. It loves to explode. Not really love to do it, but it explodes easily, right? It ruptures easily. So if someone's been in a trauma like that, you should make sure they don't have a ruptured spleen. Okay, so the fat stored under the skin is mainly to keep you warm, keep heat transfer down, because it doesn't conduct heat very well. And the stored visceral fat around those internal organs is to keep you from mechanical stress, from tearing or injuring those organs. Okay? So let's take those fats, move them out of those fat cells and mobilize it and get them ready for degradation so we can use it as fuel. Right? So that's called lipolysis. Right? We're going to take the fats, lipids, and do lysis or breaking of those molecules, so lipolysis. So we're going to take them, put them on a carrier and send them to another cell, probably to the liver. Right? Lots of cells do this, but mainly the liver. And we'll break it down into its constituent parts. So our triacylglyceride you see on the right has 3 ester bonds. We can use a lipase, right, which is a hydrolase. It uses water to break those bonds. So 3 water molecules come in. We did this in another chapter. And you end up with the 3 fatty acids and your glycerol. What do we do with those things? Well, let's see what happens to the glycerol first. It's a rather small molecule. Right? So that small molecule at the bottom, glycerol, is almost ready to enter glycolysis. We'll get to the top picture in just a second. But at the bottom, you have glycerol. We need to do a couple things to it, and it's ready to become a molecule in glycolysis, or gluconeogenesis. So glycerol, it's got 3 carbons and 3 hydroxy groups. If I use ATP and glycerol kinase to put a phosphate on one of them, because that's what kinases do, they add phosphates to things. So it transfers it from ATP onto the glycerol. You have a glycerol 3-phosphate. Wonderful. Almost there. We're not quite at one of your molecules from a glycolysis. But if we were to oxidize one of the other OH groups, all right, how about the one in the middle? Let's oxidize that middle OH from a secondary alcohol to a ketone. All right, you're going to use NAD+ to do that. And it's going to form dihydroxyacetone phosphate. And if you remember, that's one of the products of our step 4 of glycolysis when we did the breakdown of the 6-carbon doubly phosphorylated fructose into 2 3-carbon singly phosphorylated molecules. And I could convert it to the other aldehyde form if I need. So if I take 2 glycerols here and do this, I could store this as a glucose if I wanted to. Or I could send it down the pathway and end up at pyruvate. Right, so that's rather simple. What happens to the glycerol? It's the water-soluble part of it. But what happens to the lipid part of it, the fatty acid I took off? Right, that's not so water-soluble, so we need to see what we do with it. But before we do, at the top, how does a cell know when to do this? Right, it just doesn't do it randomly. We want to store it as triacylglycerols, or we want to break them down and mobilize them. It needs some instruction. So one cell is going to tell another cell to do this. And to do that, we send a signal from one cell to another. These are called hormones. Right? This one happens to be a very small hormone called adrenaline or epinephrine. All right? It binds on the surface of the cell up there. The hormone's a little blue plus sign represented in this diagram. It binds to this little receptor. It sends a signal across the membrane. That's called signal transduction. We can do a whole lecture on that another time if you want. But the gist of it is it does a cascade of events inside the cell where we turn ATP into cyclic AMP. Cyclic AMP activates a protein kinase called protein kinase A. So it goes from red to green in this figure. It means it's active now. And that protein kinase does what kinases do, and it phosphorylates things. So it phosphorylates these lipases we were just talking about that cut off the fatty acids from the glycerol. And that's all this is showing. Do you need to memorize this cascade? No. The idea here is to remember that one cell can communicate instructions to another cell to start mobilizing these fatty acids. Okay, so we saw what happens to the glycerol. Let's see how we deal with the fatty acid at this point. So on the figure at the top, glycerol simply becomes the component that can enter glycolysis or gluconeogenesis. And you can either make pyruvate or glucose from that. Fatty acid, however, can't easily travel through the blood. Right? The glycerol can very easily. It's water-soluble, very small. But the fatty acid is a big, long carbon chain. It's not very water-soluble. So to get it from the fat cell to the liver cell or other cells, wherever we're going to do it, I need to link it to a taxi, right? A water-soluble protein called albumin. And we have lots of albumins. It's a very common taxi in the blood. And it carries our insoluble thing to another cell. It drops it off at the cell surface. Right? It goes inside the cell, and then we need to get that fatty acid into an area where we can do degradation. So fatty acid degradation in eukaryotic cells takes place in the mitochondria. Right? Fatty acid synthesis will be in the cytoplasm, so we'll do that later. But we want to break these down. We need to get them not only inside that cell, but inside that cell's mitochondria. So I need to carry it over there. Once we do the oxidation or the degradation, which is what the rest of this talk is going to be about, we make a bunch of acetyl-CoA. And you already know from a couple lectures ago, acetyl-CoA enters the citric acid cycle and gets oxidized to CO2. Right, so let's see what we do with that fatty acid oxidation part. That's the only piece that you don't know yet. Okay, so to get started, we need to take our raw fatty acid, which is represented by the green molecule on the left. It could have any number of carbons. It's usually an even number. So, we have 20 carbon fatty acids, 16 carbon, 12 carbon, so forth. There are some odd ones out there, but we'll not talk about them too much. But most of them will be even. And it's represented by that thing, R could be any length chain, right? It's just a long chain. And at the very end is a carboxyl group, which makes it a fatty acid. It's a carboxylic acid. Right? We need to attach that to a CoA molecule, right? We talked about CoA before when we were doing the citric acid cycle. It's just a carrier of carbon chains. So that's what we're going to use it for. But to put this carboxyl group on CoA is going to require an input of energy, right? It just won't hop on its own. It's going to make an unstable bond. Right? So we need to use the energy of ATP to put this thing on. Now remember, ATP isn't just this ball of energy as some biology textbooks will represent it. It's an actual molecule. It has some high energy phosphate bonds in it. So how do we get from the molecule on the left, the fatty acid in green, plus CoA, to the molecule on the right, which represents the fatty acid attached to CoA through that thioester bond? And if you remember, thioester bonds are not incredibly stable. So the first thing we're going to do is take the fatty acid and attach it to the ATP molecule, right? And that's shown on the next slide, right? So here in the first reaction, we have the fatty acid plus the ATP, and unlike kinases, which permanently borrow the phosphate from the ATP to phosphorylate something, here we're interested in the A part of it, not the phosphate so much, but the adenylate, right? The sugar and the adenine. So I'm going to take the ATP, attach it to the fatty acid, and let the last two phosphates go. That's the PPI, the pyrophosphate. Recall, ATP has the adenine, like you see in the top right there, the adenine, the ribose sugar ring, and then 3 phosphates. We've lost 2 of them, leaving 1, and we attached it to our fatty acid. That's called an acyl adenylate. Adenylate for the part in red there. And then the green part is the acyl, which means a fatty acid that's attached to something. It's now an adjective, like you learned your substituents. Okay? In the next reaction, we take that very product, that acyl adenylate, and CoA comes along, and the sulfur of CoA bonds to the carbon that's double bonded to the oxygen there, displacing the AMP. Kind of takes its place. So one comes in and the other leaves. Both of these two reactions, the first one, number 1, and number 2 there, are easily reversible. They don't go back and forth all the time. So how do we get it to go forward? Well, in the first reaction, we release the molecule of pyrophosphate, that PPI sitting there. Right? Pyrophosphate is just 2 phosphate groups attached to each other. It's an anhydride bond, or anhydrous bond. And you guys remember, if you did this in lab, if you get anything that's anhydrous wet, it tends to break the bond using water. Right? It's a very spontaneous, favorable reaction. So in the cell, that happens, it breaks the bond, but it's just not fast enough for the cell. So the cell has an enzyme that also breaks this bond even faster. Right? It's called pyrophosphatase. That makes sense because it breaks pyrophosphate. And it degrades it into 2 individual phosphates. Once I do that, reaction number 1 can't go backwards. So this is Le Chatelier's principle. If I remove a product, it tends to go in the direction that replenishes that product. Right? If I disturb equilibrium, it tends to go in the direction that will restore equilibrium. So I'm removing a product, which allows me to go on to the next step, because it can't go backwards, and force it to become acyl-CoA. Again, I'm supplying more fatty acid at the beginning. I'm going to be removing the acyl-CoA at the end. So overall, I'm also importing a Le Chatelier principle, adding more raw materials and removing the product. So it's going to go forward. What did this cost me? Let me go back a slide and show you that. It cost me 1 ATP molecule, yes. But in terms of how much energy or how many high energy phosphate bonds did it cost me, we're going to say it cost 2. Because I did remove 2 phosphates from the ATP, right, as pyrophosphate. So in terms of the energetic cost of this reaction, I know I'm using a single ATP molecule. But think of the ATP molecule as a debit card. I'm going to charge it twice. Right? I'm going to take 2 phosphates away from it. So in the end, when we tally how many ATPs in energy this costs, this one's going to count as 2 because I charged that ATP card twice. Most of the time we think of it as only once, but in this case we're charging it twice. So far I'm in debt for 2 ATP equivalents, I'm going to call it, or 2 high energy phosphates. Right, so I'm not getting any energy out of this yet. I'm in debt for 2. This should sound familiar. We did this back in another pathway. Right, so I have my acyl-CoA. I have my green fatty acid at the bottom attached to CoA. Good, it's ready to start the reaction. And so for the next part, I need to get that molecule inside the matrix. So I put it on CoA, but if you remember, CoA is a very big molecule. It's got a lot of charges. It's huge. It can't go through the membrane easily. So I need to get it across the membrane into the mitochondria, but I can't force the CoA to go through. So we have this little translocase to help me get this through. It allows the acyl group, the green carbons, to get across the membrane without having to drag the CoA through. Okay, so the pools of CoA in the cytoplasm and in the matrix don't have to exchange. So here's how it works. The acyl-CoA, as you see on the left, relieves itself of its CoA group, right? Just the fatty acid gets put on a molecule called carnitine. And it's called carnitine because it sounds like carnivore. And if you find this molecule in virtually every piece of muscle you'll ever eat, right? Muscle or meat or something, because they're loaded in mitochondria, right? And you need this to get your fatty acids into mitochondria. So it's difficult not to get carnitine in your diet. Okay, so we take our fatty acid off of CoA, put it on carnitine. That makes an acyl-carnitine, and our CoA is free to go. It can go pick up another if it likes. So this molecule is now transferred onto a carrier. That carrier goes across the membrane. As you see on the right, the acyl-carnitine goes down into the matrix, and at the same time, just a raw carnitine comes out. So this is an antiporter, one in, one out. And while inside, almost identical reaction happens in reverse. We take the fatty acid off of the acyl-carnitine and give it back to a green CoA in this case, another CoA that's in another compartment. So overall, what have we accomplished? We took our acyl-CoA and moved the acyl part of it inside the matrix without having to move the CoA part. Right? So this accomplishes getting the carbons in, but it bypassed having to drag the CoA through the membrane, right? Because it's not very easy to get that through the membrane. And that's all this is doing. So at this point, we've taken our fatty acid, activated it by putting it on CoA, then we translocated across the inner mitochondrial membrane into the matrix of the mitochondria, so the inner part, and we're ready to start oxidizing it, degrading it. Okay, so that's the next steps, and it's 4 steps that happen over and over and over, and I have a little acronym for them, right? You can remember them by the term Ohio State. Okay, so if you look at Ohio State in the letters and kind of replace the I with a Y, I guess, it's oxidation, hydration, oxidation, and then sulfur transfer. Right, so O for oxidation, high if you spell it with a Y, I guess, for hydration, O for oxidation again, and then the ST for sulfur transfer. Okay, more descriptive, what's actually going on here is in the first step, we're going to oxidize that acyl-CoA. We're going to use the cofactor FAD to do that. We'll explain why FAD in a minute. Once we do that, we'll have a carbon-carbon double bond, and I'm going to show you pictures of this. With that carbon-carbon double bond, we're going to do something you're already familiar with. We're going to hydrate it with water, and if you remember, we take an alkene, and you hydrate it, it becomes an alcohol. So we'll end up with an OH group on one of these carbons. Then we'll take that alcohol, which will be a secondary alcohol, and oxidize it using NAD. Why NAD? We'll get to that in a second. And a secondary alcohol oxidized will become a ketone. Okay, so we'll have a carbonyl on there. And then finally, we'll break that carbon-carbon bond between 2 and 3, or alpha and beta, using CoA. So CoA comes along and attacks the same carbonyl we just made, breaking the bond, and I have 2 pieces. Okay, so let's show that pathway in the next slide, but I want you to remember our numbering system here. I'm going to refer to it by number or by symbolic notation, alpha and beta, gamma, delta, and so forth. You got to know which one's which. So if you remember your nomenclature, if we have a long carbon chain and you wanted to name it, the first thing you want to do is find the longest continuous carbon chain with the highest priority in mind. Well, this is a carboxylic acid. That's going to be number 1. That was at the top of your list. So number 1 is where the carboxyl group is, followed by number 2, 3, 4, 5, all the way down to however many it has, 12, 16, 20, whatever. We also talked about this when we did fatty acid descriptions, the same symbolic notation. Carbon number 1 doesn't get a symbol. It's called the carbonyl carbon. Carbon number 2 is alpha, 3 is beta, 4 is gamma, 5 is delta, and so forth. Right? You also may remember we called the last one omega. Right? Won't come into play too much here. We're going to do all our work down at this end where the alpha and betas are. Okay, so if I refer to carbons 2 and 3, which is where all the action is going to take place, I may also refer to them as alpha and beta. All the action is going to take place at this alpha-beta connection. Okay, so our first step, remember, Ohio State, first step, oxidation. So I have my acyl-CoA at the top left of the figure. How many carbons it has, I don't know. I just have an R at the end. It's a very long chain. We're only going to deal with the beginning. Right? So all the action here is taking place between carbons 2 and 3. That's the ones in red in the diagram. So the alpha and beta carbon respectively. So I'm going to oxidize it. What am I doing here? I'm really taking away a couple hydrogens and their electrons, right? Mostly the electrons, the protons go along for the ride, and I end up with a double bond between those two carbons, right? And if you had to name this thing again, you just tell me that it's got a double bond at position 2. And that's why you see the little delta 2 in the name. You should be familiar when we did that. Why did I use FAD and not NAD, right? I could have used NAD plus or FAD. Both of those are excellent cofactors for doing oxidations. Here's a little general rule that will almost never fail you, right? So you can almost always rely on this. And for everything we're going to do, absolutely rely on this. So when you have an oxidation reaction like this, and you're taking away those electrons, right? And the hydrogen protons go along with them. From whom am I taking them? If I'm taking the electrons from an O, N, or F, oxygen, nitrogen, or fluorine, remember those are the 3 most electronegative atoms. They tend to not want to lose their electrons. They like them. They're electronegative. They love their electrons. They have a high affinity for their electrons. So if you're going to take it away from an O, N, or F, you need to use NAD+. If you're not taking it away from O, N, or F, then I'm happy to use FAD. I don't need the NAD. I can just use FAD. It's sufficient. It's actually better. Now, I don't mean 2 O's, 2 N's, or 2 F's. If either one of the 2 molecules or 2 atoms is an O, N, or F, I need an NAD. And we'll see that in a few steps. Here I have 2 carbons. Neither is O, N, or F, so FAD will work just fine. And that rule will never fail you for what we're going to do. Okay, so I start with the acyl-CoA. I'm taking away those electrons and the protons that go with it using FAD, because I had a pair of carbons there. I end up with my double bond. So that's the first step, oxidation by FAD. The next step, we turn that alkene we just made into an alcohol. We're going to hydrate it. You guys remember we did this with Markovnikov's rule, right? Where you add an asymmetrical molecule to an asymmetrical double bond, and the side with the most hydrogens gets the hydrogen, and the other side gets the non-hydrogen thing, in this case the OH. Well, enzymes kind of ignore that rule and do what they want, right? So they're specialized to put the OH in the same spot all the time. So in this case, the enzyme that does this step always puts the H on carbon 2 and the OH on carbon 3. Or another way to say it is puts the OH on the beta carbon and the H on the alpha carbon. Okay, that's shown in the figure where the blue OH ends up on carbon 3 or beta and the blue H ends up on carbon 2 or alpha. So I've turned my alkene into an alcohol. Right, and then the last step we have, well, not last step, but last oxidation step. We did O for oxidation, high hydration, and then O is another oxidation. This time I'm going to take away the H on the OH, right, of the OH, and then the H of the other side, one of those 2 hydrogens on carbon 2. Oh, sorry, one of those hydrogens, the only hydrogen on carbon 3. So you can't see my mouse, I know, but it's the H that is part of the OH and the H on carbon 3 as well. That's going to end up putting a double bond between the oxygen and carbon. Right? So for some reason, the oxygen turned red in this picture, but that's okay. Right? But we now have a carbonyl there. So I went from secondary alcohol to ketone, right? Or carbonyl. Why am I using NAD plus here and not FAD? Well, one of those 2 things was an O, N, or F. Right? I took the hydrogens from an O, N, or F, at least one of them, so I will require NAD plus. So I get another NADH out of this. At the bottom, I have a ketone group because we turned a secondary alcohol into a ketone, right? And the ketone's on the number 3 carbon, 1, 2, 3. also known as the beta carbon. So I have a beta keto acyl-CoA. That is beta keto arrangements are inherently unstable. The alpha ketos, very stable. Beta ketos, inherently unstable. So we need to relieve this instability. So now our last step, the state part of it, or ST, or sulfur transfer, or thiolysis, we're going to have a CoA come in. The CoA sulfur is going to attack this beta carbonyl, right, the pink one there. And we're going to break the bond between carbon 2 and 3, the pink carbon-carbon bond. When I break that bond, right, the carbon on number 2 will grab another hydrogen, usually from the CoA that's losing a hydrogen here, and it will take off as a 2-carbon CoA. That's known as acetyl-CoA, shown on the far left at the bottom. What's left looks identical to what I started with, except it's been shortened by 2 carbons. Now, I don't mean 2 carbons I've removed from the tail end, not the omega carbon. That was never touched. I mean carbons numbers 1 and 2 are now gone. They've been cut off. And carbon 3 has become the new carbonyl carbon. Carbon 4, right, over here, has become the new alpha carbon. Everybody got promoted by 2 ranks. So if you were the beta carbon before, this one, you are now the carbonyl carbon. If you were the gamma carbon, or carbon number 4, you're now the alpha carbon, or number 2. Right? If you were the 15th carbon, you're now the 13th carbon. Everybody got promoted by 2 ranks. Okay? And carbons 1 and 2 left as acetyl-CoA. So to review this whole pathway again, what do we get? We start with a long tail attached to a CoA, right? An acyl group attached to a CoA. We do an oxidation, gives me a double bond. And out of that, I get an FADH2. And we already covered what happens in the electron transport chain. That can be used to pump some protons. So I can get some energy out of that. And then next step, I hydrate it. I don't get or lose anything in this step. I just make an alcohol. But I can now oxidize that secondary alcohol and get an NADH out of it, which again I can use in the electron transport chain to pump some protons and get more ATP made. And then finally I get my CoA to come along and cut off those 2 carbons to relieve that unstable beta keto or 3 keto arrangement. And I get an acetyl-CoA out of this. You know the acetyl-CoA can enter the citric acid cycle because we said we're doing this in the matrix, so that's where that takes place. And that can enter the citric acid cycle and again generate more NADHs and FADH2s to generate more ATP. Okay, so I get a lot of potential ATP made from doing this. And my fatty acid acyl group has been shortened by 2 carbons. But can't I do this again? It's still, if it started out as 20 carbons, it still has 18 carbons there. I can do this over and over and over until I run out of carbons. So that's what we're going to do. We're going to repeat these same 4 steps until we run out of carbons. Okay, that's shown here on the right. So we'll get to the numbers and such on the left in a second. But let's look at the molecule at the very top. Right? And if your figure doesn't look like mine, I modified this about a half hour ago to show you all the steps. It was only showing the first 4. I'd like you to see all of them. So I'll give you the new figure as soon as we're done. So I took the similar figure and modified it and let you count the steps we're going to do. So at the very top, if you count those carbons, there are 16 carbons there. I know it doesn't look that long, but we have a little indication here that there's 7 CH2s, and then finally we end with the CH3. Yes, they could have used a number larger than 7 if they included all 6 of these as well, but they wanted to show these. So there's 16 total carbons here, and you see all the action is going to take place down at this end. Okay, so in the first round, what do? We do oxidation. We're going to put a double bond right here, right? Then we're going to do hydration of that double bond. We're going to end up with an OH at this beta position. We're going to do oxidation again with NAD, and we end up with a carbonyl here. And then CoA comes along and attacks that, right? It's going to be a blue CoA in this case. Blue CoA comes along, attacks here, and everything in red leaves as acetyl-CoA. This beta carbon is now the new carbonyl carbon. And everybody's been promoted by 2 ranks. That's round 1. What did I get out of round 1? I got an FADH2. I got an NADH. I did consume a water molecule, but we're going to ignore that because it doesn't really hurt anything or help anything. But I did get an acetyl-CoA. Right? So I got 3 main products out of this. 1 FADH2, 1 NADH from the 2 oxidation steps, and I got an acetyl-CoA that was cut off. So now what this thing has 14 carbons. Let's play our game again. I make a double bond here, right? I oxidize it with the FAD. Then I add water to hydrate it. This is now the beta position, so it gets an OH. Right? Then I oxidize that to a carbonyl. I add this time it'll be a green CoA that comes in and cuts this carbon-carbon bond here, releasing yet another acetyl-CoA. And now my chain is only 12 carbons long. Right? So it's been shortened by 2 more. We do the exact same thing again. I just want to show you all the steps. And we end up with one that is only 10 carbons long. So far I've released 3 acetyl-CoAs. I'm sorry, the color goes away here because I was just copying and pasting. But I go from 10 carbons, right? This is 7, 8, 9, 10 carbons, down to 8 carbons, 5, 6, 7, 8. And I've released a 4th acetyl-CoA. But we're not done. This thing has more carbons. So if we continue, I put that guy at the top with its 8 carbons. And our 5th round, we get yet another acetyl-CoA. And then I drew it out. We're down to 6 carbons again. Right? Let's continue. We do another round. I make a double bond here. I put the alcohol group there. I oxidize it to a carbonyl. I break this carbon-carbon black bond. Get another acetyl-CoA, and I'm down to 4 carbons. I've done 6 rounds. I've got 6 of everything so far. I do it yet again. So here I put a double bond between the blue and the green carbons. I add water to make that an alcohol. I oxidize it using the NAD plus to make the carbonyl. And I release it with I use the green one then. A green CoA comes in and cuts off this blue acetyl-CoA. Leaving behind, we went from 8 to 6 to 4 to 2 carbons left. But I don't need to do an 8th round because look at my final product. It is an acetyl-CoA. It's only 2 carbons long. So for a 16 carbon fatty acid, I don't do 8 rounds. I do get 8 acetyl-CoAs, but I only did it in 7 rounds. So if you really think about it, the number of carbons, let's play a different one. Let's not pick 16. Let's say we only had 10 carbons. How many rounds would you do with 10 carbons? Well, you wouldn't do 5. You would get 5 acetyl-CoAs. That's correct, because that accounts for all my carbons. But you're only going to do 4 rounds with 10 carbons, because each round you remove 2, and after that 4th round, the final residual piece is an acetyl-CoA. So in our case of 16 here, we'll do 7 rounds, which I laid out for you. We'll get 7 FADH2s produced. We'll get 7 NADHs produced. We'll consume 7 water molecules, of course. But we'll get 7 acetyl-CoAs, 1 from each round. And the leftover piece is our 8th acetyl-CoA. So a total of 8. 7 from doing the rounds, and then my leftover piece makes 8. Don't fall into an algorithm of just memorizing numbers. Make sure you know how this works. Okay? So overall, how much ATP do I get out of this? We said we're going to use these NADHs and FADH2s and acetyl-CoAs in our citric acid cycle and electron transport chain. So how do we get it? Let's tally this up. So each acetyl-CoA, if it were to go through the citric acid cycle and the products down the electron transport chain, is roughly worth 10 ATP. And each FADH2 is roughly worth 1.5 or so ATPs. And each NADH is about 2.5. So if you take all those numbers together, I have 8 acetyl-CoAs worth 10 each, that's 80. I have 7 FADH2s worth 1.5 each, that's 10.5. This is very easy math, right? 7 NADHs worth 2.5 each, multiply, you get 17.5. If I add these 3 up, I'll get 108. Then I subtract 2, then I get 106 total. Why did I subtract 2? Well, if you remember, go back a few slides. I grabbed a picture and threw it down here for you. When we started this thing, seems like a long time ago, we had to spend that ATP debit card twice. We charged it for 2 ATPs, 2 high-energy phosphates, to put the thing on CoA. Remember that? So it's much like glycolysis, where yes, you get a total of 4 ATP produced in glycolysis, but we had to spend 2 initially to get things started. Same thing here. We get 108 out of this, but I had to put 2 in to get it started. So my net is only 106. So I'll leave this as a question for you guys to do on your own. Which is a better food source in terms of the total amount of ATP per carbon? Glucose or fatty acids? So how do you answer that question? Glucose has 6 carbons, right? And if you go through all the pathways of glycolysis and pyruvate dehydrogenase, citric acid cycle, electron transport chain, ATP synthesis, and so forth, if you do all of that, not taking into account any losses, you'll get roughly 32 ATP out of it. So 6 carbons of glucose gives you 32 ATPs. This 16 carbon fatty acid gives me 106 ATP. Of course, there's more ATP here. It's a bigger molecule. It's got more carbons. But per carbon, which one is better? And I'll leave that for you to figure out on your own. Okay, so everybody gets the process of how to break this down. If I give you a different size fatty acid, not 16 carbons, but maybe 20 or 12 or 14, it'll be an even number, right? And you can tear it apart and tell me how many you get. Okay. What about some unusual fatty acids? What if they don't have this nice long tail with no double bonds in it? What if they already have a double bond in it? We talked about some unsaturated fatty acids. They had the delta nomenclature with the numbers on it, like a arachidonic acid. You don't have to memorize this one or the name. It had 20 carbons and 4 double bonds in it. So when you learn how to write it with the colon 4 delta and the right positions, we didn't talk about how we degrade those yet, and we won't for this class. All I want you to know for that is I need to deal with those double bonds. So when it happens to be in an odd position, like in numbers 3, 5, 7, 9, and so forth, it's in the wrong spot to do this reaction. I need to move it to an even position. So just moving a double bond, you could probably guess it's an enzyme that's an isomerase. So I'm going to need an isomerase if I have double bonds at odd positions. What if they're at even positions? Well, it seems like it would be perfect. It's already where it needs to be. Except there we run into a problem because we're going to make a very stable conjugated alpha keto system, or alpha arranged system. So we're going to have to reduce it, which is going to cost me something. So I don't want you know all the pathways of that. If you really want to know it, go watch the other video that I did from my senior level biochemistry class. But I want you to know that if we have a double bond there, we're going to need an enzyme called an isomerase to move it if it's in an odd position. And if it's at an even position, you're going to need the isomerase and a reductase to take care of the stable arrangement we're going to get. Okay, so I wrote that in the slide here. Know that we're going to need an isomerase and or an isomerase and a reductase. Right? We need both if it's at an even position. Odd, you only need the isomerase. Okay. Right, moving on, we've degraded all our fatty acids into acetyl-CoA. We see how much ATP we can get out of it. We're finished with it. What if you're trying to take acetyl-CoAs and not degrade them and use them for fuel, but to store them some other way? Because acetyl-CoA is not incredibly stable. It's got that thioester bond. We want to store it another way. So we can either store it as fatty acids, which we'll save for the end, or we can store it as ketone bodies, right? So the problem with here is ketone bodies. When would you do this? Well, I can think of 3 situations where you need to make a lot of ketone bodies, right? So if you think back to our citric acid cycle, what 2 molecules did we use to start the cycle? Well, we had acetyl-CoA coming in that we just made, and we had an oxaloacetate that was there. Again, you can make that from pyruvate as well. Right? We had an acetyl-CoA and an oxaloacetate, both made from pyruvate, entering the cycle to become citrate, and we continue through the cycle. I need a balance of those 2. One for each. One acetyl-CoA and one oxaloacetate. So what if you don't have a balance of those? What if you have an imbalance? What if you have much more acetyl-CoA? What if you have much more acetyl-CoA than you do oxaloacetate? So how is that possible? All right, someone's got their microphone on. So what if your balance of acetyl-CoA to oxaloacetate is not correct? You have far more acetyl-CoA than you do oxaloacetate. How can that happen? Since they're both made from pyruvate. Well, if you're making them from pyruvate, that won't happen. But what if you have an influx of a lot of fats that you eat? Right? So your diet is loaded in fat and not a lot of sugar. Okay? So that's the case in these keto diets. Right? So they're eating lots of fats and proteins, but very little glucose. There are other sugars there, but very little glucose. So there's an imbalance. There's much more acetyl-CoA than there is oxaloacetate. So what your body's going to do is, instead of using the acetyl-CoA and oxaloacetate 1-to-1 to keep doing this, you're going to have an excess. So I need to take the acetyl-CoA and store it for later. So your body's going to make ketone bodies. Okay? Or it's going to store it as fat in the other cases I'm going to mention. So the other cases, such as, let's say, alcoholics. Alcoholics tend to eat things as well. They don't have a lack of glucose, but they do have an overabundance of ethanol. Right? So the amount of ethanol is far in excess of the amount of sugar. So the ethanol, being two carbons, gets converted to acetaldehyde and then ultimately to acetic acid. And then the acetic acid gets put on CoA to form acetyl-CoA. So we have an overabundance of acetyl-CoA again. What's going to happen here is, since there isn't a lack of sugar, plenty of sugar in the diet, we're going to take the excess acetyl-CoA and store it for later. So we're going to turn it into fat in the liver. So alcoholics tend to get fat. There's a lot of fat in the liver, and the liver will eventually become fibrotic and then cirrhosis and fall apart and die. So the other situation where you have a lot of acetyl-CoA compared to sugars is uncontrolled diabetics. So in the case of a diabetic, there's lots of sugar, yes, but it's in the blood. From the cell's point of view, there's very little sugar around. It can't get in the cell. So from the point of view of the cell, an alcoholic is a lot like an uncontrolled diabetic. There's just not any sugar coming in compared to the large amount of acetyl-CoA coming in. And so a fat cell can mobilize a lot of fats, and cells are abundant in acetyl-CoA, but if you're a diabetic, there's a lot of glucose coming in. You can't make a lot of pyruvate or a lot of oxaloacetate to go with it. So again, we're going to store it as fats, or we're going to make ketone bodies. And then the last situation is just simply during starvation, right? I think I said 3, I might have mentioned 4, right? But during starvation, you're getting no intake of food whatsoever. So when your glucose supply is limited after, say, day 20 or 30 of starvation, assuming you have water, at this point, you're feeding off of your own molecules, right? Your proteins are being degraded, your fats are being mobilized, all the glycogen's gone at this point, and you have an excess of acetyl-CoA compared to sugar again. So on all these cases, you're going to make a lot of ketone bodies. So the ketone bodies will be high concentration in the blood, and you can check that in starvation, in untreated diabetics, in alcoholics, or if you're doing this keto diet, which is the point of the keto diet. Okay, so let's look at the ketone body formation and how we make it from acetyl-CoA. So starting at the bottom left, we have 2 acetyl-CoA molecules, right? One in red, one in blue. And they really don't want to interact with each other that much because there's not a very reactive group there. But one can attack the other. We're going to say the red one at the top, you see the methyl group sticking off, the CH3. Let's say we somehow deprotonate that by an enzyme. We take one of its electrons and protons away, or 2 of its electrons and protons away, or just a proton away, and then it attacks the blue carbonyl at the bottom. So if you imagine this CH3 becoming CH2, and then attacking this blue carbonyl at the bottom, right? I'd attach this carbon to this carbon. I have a new carbon-carbon bond. That's represented here, right? At the same time, this carbon can't have 5 bonds, right? So it loses the bond to the CoA. So I make a new carbon-carbon bond, the red methyl group, which is now a CH2, to the blue carbon of the carbonyl, and I break the carbon-sulfur bond, which was already very labile in the first place. And you see the CoA leaves. And of course, it's just written as CoA, but that also represents the sulfur going with it. It's CoA-SH if you want to write it out. So now I have 4 carbons in a row, and that's called aceto-acetyl-CoA. It's an acetyl-CoA with an acetate group on it. So aceto-acetyl-CoA. Right? We play the same game again. A third CoA comes in with this acetyl group. And again, the green methyl group this time gets deprotonated. I'm going to have it attack the very same carbonyl. But because I'm attacking the same one, now there's going to be 3 carbons attached to this blue carbon. So it will be necessarily branched. Right? It can't all be in a row anymore. And when this blue carbon gets attacked by that green methyl group, we don't leave this double bond here. It becomes a single bond, and a water molecule puts an H on that OH. So that's shown over here. Right? So now I have 5 carbons in a row, right? 1, 2, 3, 4, 5, with a methyl group sticking off. Right? I also have an OH here. Right? So this OH comes from this original blue carbonyl that's become reduced to an alcohol, right? And we protonated it with a water molecule. And at the same time, we also take the rest of the water molecule, the OH minus, and cut off this green CoA, right? Leaving behind just the carbonyl. Okay, so I have a 5 carbon molecule still on the original CoA, and now it's branched. It's got a methyl group and an OH group. And if you count the carbons, this is number 1, 2, 3, 4, and 5 because this is the most important end where it's attached to CoA. On number 3, I have a methyl, and on number 3, I have a hydroxy group. So that's its name. It's called 3-hydroxy, 3-methyl, glutaryl-CoA. Glutaryl means 5. Right? So if I have 5 carbons in a row, there's a carboxyl group on each end. We call it glutaryl or glute. If you look back at your amino acids, you'll see that with glutamate and glutamine as well. Right? Or alpha-ketoglutarate all had 5 carbons with this pairing of carboxyl groups. And we have a methyl and an OH on number 3, as indicated in the name. Finally, we have the original CoA in red with its acetyl group just leaves, right? That allows me to to collapse this OH back into a carbonyl, right? So this H leaves with that carbon, right? This forms a double bond again, and I'm at acetoacetate. And there's no CoAs left on this thing. So this is one of my storage forms of acetyl-CoA. I've really effectively used 3 of them to put 2 together. Right? And this is all going to be one direction. You really don't go backwards when you're making this. You can, but it generally goes in this direction if you're making it. And acetoacetate, again, is not stable. It's because it has this beta-keto arrangement one more time. Right? So this would be carbon number 1 now at the bottom. For 1, 2, 3, 4 carbons. And on number 3, or on beta, is a keto. So I have a beta-keto acid one more time, so not stable. The simplest thing this can do is just lose number 1 carbon as CO2. And if this were to leave as CO2, that leaves behind acetone, which is not a good idea. So if you're making ketone bodies in your blood, you don't want a lot of acetone floating around in the blood. It's an organic solvent, it's going to disrupt membranes, and cells are going to die. So let's try not to let that happen. To also resolve the situation in a safer way, we're going to take this blue carbonyl, right, this ketone at number 3, and reduce it down to a secondary alcohol. But I'm going to need an NADH to do that. You notice not NAD, but NADH. This is reduction, but oxidation of this molecule. Okay, so reduction of the acetoacetate to the 3-hydroxy butyrate. Butyrate is our name for 4, right? If you remember, but means 4. We'd rather do that than store it as acetone. That's a bad idea. So a lot of these molecules we're making, this acetoacetate, this 3-hydroxybutyrate, we prefer not to make a lot of acetone, is floating around in the blood. And if you're in the starvation mode, or if you're uncontrolled diabetic, or if you're the raging alcoholic, you're going to make a lot of these things. And so let's look at the starvation one. After a few days of starvation, on the chart on the bottom left, you notice the amount of glucose in the blood starts to drop. Right? After about day 2, day 3, it's not dropping much anymore. It's kind of leveling out. What's going on? So the amount of glucose, we're not making much more glucose, but we're mobilizing the stuff we talked about last time, all that glycogen storage in the liver. Right? It's being mobilized and released to maintain the blood glucose level. That can only go on for so long. Right? You only have so much store in the liver. Remember, it's like 10% of its mass. But you can't do that indefinitely. So if we keep starvation going for 10, 20, 30, 40 days, assuming you still have water at this point, you're going to run out of glucose in the liver as stored glucose, glycogen. And we need glucose from another source. Or many of your amino acids, in fact all but 2 of them, can be turned into glucose. Leucine and lysine cannot. But all the others can be turned into glucose, so you're going to start degrading proteins, mainly your long muscles, like in the back and in the legs and in your arms, because if you're starving after 40 days, how useful is that back muscle? Right? You really don't need it to survive. I keep the heart going, I keep the diaphragm and the muscles that move it going. You might want to keep the chewing muscles going in case you do find food, but you don't need that big back muscle and the really big leg muscles. So we'll start degrading those for their amino acids so we can make more glucose. At the same time, you see the amount of ketone bodies is going to spike. Right? We're going to make a lot of ketone bodies after the 2nd, 3rd, 4th day. And we're also going to start mobilizing fatty acids. Our fat cells are going to supply acetyl-CoA, but they can't supply glucose. And that's a main fact I want to get across on this slide. I can't say it enough, that you cannot turn fatty acids into glucose. You can always turn glucose into fats. That's not a problem. But you can never turn fatty acids, or acetyl-CoA, back into glucose, because we can't turn acetyl-CoA back into pyruvate. If you remember from our pyruvate dehydrogenase discussion, when we lose that as CO2, it was completely and utterly irreversible. You can't go backwards. Right? So animals cannot convert fatty acids into sugars. We can always go the other way. We can turn all the glucose we want into all the fatty acid we want, but we can't go backwards. There's only a few times that fatty acids can be turned into glucose, and that's done by certain microorganisms and plant seeds. But we're not going to cover that in this course. But as far as you're concerned, animals cannot convert fatty acids into glucose. Okay? And the last thing we want to talk about is how we take acetyl-CoA, and instead of making these ketone bodies, let's get away from the starvation model and the diabetic and alcoholic, and let's say we're in a very well-fed state, like these bears that are catching salmon up here, right? They're storing up food for the winter, right? Because they're going to spend a lot of time just lounging around, sleeping, doing nothing. So they don't need a lot of fast fuel meaning sugars. They can live off of their fat stores, right? They're not going to be getting up and chasing anything. So they're just going to be resting through the winter. So we can store a lot of food as fat, but it's a very slow release and delivery. They're very lethargic if you wake them up. However, if you want to be fast, you need sugars. So they don't need to store sugar that much. They're going to store it as fat. So they're eating a lot of foods. We're turning them into acetyl-CoA, and we want to store that acetyl-CoA as fatty acids. Okay? And the picture at the bottom left was what I was talking about in alcoholics. If you have excess acetyl-CoA compared to your sugars, right, or pyruvate, or glucose, then you're going to store those excess acetyl-CoAs as fatty acids, just like these bears are intending to do. Except they're doing it for a reason that they're going to consume it over the next few months during winter. Alcoholics aren't going to be hibernating that much. They're still going to be eating foods. So they don't need to store it as fat, but the liver is going to do it anyway. So initially it's going to store it as called a fatty liver. And that can be reversed pretty easily. Just change the diet, right? Eliminate all the alcohol, and eat regular food. If you keep going, the liver becomes what we call fibrotic, and a lot of those cells that are dying are forming scar tissues. It's still recoverable. The liver is actually very phenomenal. It can recover from fibrosis for the most part, unless it's severely damaged. But if you keep going, we call it cirrhosis. So now we have connective tissues destroying liver cells. They're not really liver cells anymore. They're just becoming connective tissue. Right? That damage is irreversible. The only way to fix that now is to get a liver transplant, and if on your resume for the liver transplant you write you're a raging alcoholic, that wouldn't be the best use of that liver. Okay? So let's talk about how we make it. So we're going to start with acetyl-CoA, and most of our acetyl-CoA is going to be found in the matrix of the mitochondria, because that's where we just made it, or that's where it's located from our degradation pathways. And we need to move that acetyl-CoA, much like we did with our acyl-CoA. We need to move this one outside into the cytoplasm, right? So, this should make logical sense. If you're going to degrade fatty acids, and you're going to also be able to make fatty acids, you probably don't want those two pathways competing with each other, because that would just be wasteful. So one way to control that is to put one in one room and put one in another room, right? So make your mitochondria the place where you degrade fatty acids, and make your cytoplasm the place where you build fatty acids. And that way you're not confused on what to do where. Right? So our acetyl-CoA is in the mitochondria. We need to move it out to the cytoplasm, and we have another little shuttle trick to do that. It's not the same one, but it's very similar. We take our acetyl-CoA, as you see in the figure in the top, combine it with oxaloacetate to make citrate. And some of you are probably thinking, hey, that's step one of citric acid cycle, and you're absolutely right. We're not going to continue the citric acid cycle here. We're just going to use that enzyme. So we'll make citrate. And if you remember back to our ATP synthesis lecture, at the very end we had all those toll booths, a very specialized toll booths that let certain things in if you give me something else out. This is one of those toll booths. If I let a citrate out, I must let a pyruvate in. So I'll take advantage of that shuttle. If a citrate goes out, pyruvate must come in. Well, I could convert the citrate out there back into oxaloacetate and acetyl-CoA. It's basically step one of citric acid cycle run in reverse. And there's an enzyme in the cytoplasm that prefers to do that. So we make the acetyl-CoA. The oxaloacetate is then turned into malate. That's also like our citric acid cycle run in reverse. And we take malate, which has 4 carbons, and turn it into pyruvate, which has 3 carbons. So we're going to lose a CO2 there. That's the malic enzyme, which we didn't talk about too much. Pyruvate goes in and can be turned into oxaloacetate by pyruvate carboxylase. We did briefly mention that one. and then we can continue the cycle. So this does cost something, right? But I do get some things out of it. I get my acetyl-CoA transported to the cytoplasm. Why am I okay with paying for this? If I'm in the mode of making fatty acids, I'm well fed. I've got lots of ATP. I'm not starving. I'm storing stuff for later because I don't need it now. So I don't mind spending a little fuel here, a little CO2 and ATP, right, to store stuff for later. Right? When the cell is well fed, it's not in conservation mode. It's let's store some of this for later. I don't care if it's a little wasteful building it. It's better than throwing it away. Okay, so the first step at the bottom, once we get our acetyl-CoA into the cytoplasm, is we need to activate it, right? So we take our acetyl-CoA and we're going to put a carbon on it, right? Put a CO2 on it. This is done by acetyl-CoA carboxylase. That makes sense because we're carboxylating it. And that makes the molecule malonyl-CoA. Okay, so acetyl-CoA, you see, has 2 carbons attached to the CoA. And when we're done, malonyl-CoA has 3 carbons. So we've added a CO2 onto the end of it. This is not an easy reaction to do. It's actually not favorable at all because I end up with, you may recognize, a beta-keto arrangement down here, not stable. And we'll take advantage of that in a few minutes as well. So to put it on, I need to get this CO2 in the form of bicarbonate, shown in red here, right, which is readily accessible in the body. We have lots of bicarbonate around. It's a buffer in our blood. And we're going to use ATP once again to activate it. Now, don't think of using ATP as just some currency that you spend, like a coin or a bill, but we're going to use it in the very similar way that we did before. We're going to take the ATP molecule and use part of it, right, a phosphate, and attach it to that bicarbonate. That makes carboxyphosphate, not stable. And then the acetyl group, again, the methyl group on the far end, like we did with ketone bodies, is going to get deprotonated and attack that bicarbonate, releasing the phosphate I just added. Okay, so it's very similar to our ketone body formation, except we're specifically putting this one on and leaving it on CoA. Okay, so in this step, what happens from the previous slide to this one, it's very subtle. I didn't really have a slide for it, but it's number 3 up here. If you look back, we have malonyl-CoA. I have 1, 2, 3 carbons on a CoA, and that same molecule is now represented here in red as malonyl ACP. Well, where'd the CoA go? What is this ACP thing? It's an acyl carrier protein. It's just a protein shown at the bottom here in blue, and it has a business end which looks exactly like the business end of CoA. So all I've done is take my 3-carbon malonyl group, plucked it off of CoA, and attached it to its cousin ACP. It's kind of a free transfer, right, from one sulfur to another. Their business ends look identical. Okay, so I'm just transferring it to a protein to do the rest of this. The protein, think of it as it's like a lifeguard that's going to carry it through the rest of these reactions, right, to make sure it does these reactions. Okay, so I've taken my acetyl-CoA, carboxylated it to malonyl-CoA, and then plucked it off of its CoA to put it on ACP. If I also start with another acetyl-CoA, don't carboxylate this one, just pluck it off and put it on a separate ACP. So if you imagine I started with 2 acetyl-CoAs, one of which I carboxylate to malonyl-CoA, and then I pluck each of them off of their CoAs and put them on separate ACPs, I'll have the situation at the top. 1 acetyl ACP and 1 malonyl ACP. Of these molecules, or of the atoms in these molecules, this last carbon on the malonyl ACP, the very last carboxyl group, is the one I just put on. Right? And that's the same one that's about to leave because it's beta-keto again. Remember, it doesn't want to stay. So I'm going to let it leave. When the CO2 leaves, it's a decarboxylation that leaves behind a CH2, which will happily attack the carbonyl in blue. This looks very much like ketone body synthesis. The only difference being it's held by these carrier proteins, this big blue thing, right? So it attacks this here, and again, the ACP is willing to leave. Right, so ACP leaves, and I end up with 1, 2, 3, 4 carbons in a row. This looks exactly like aceto-acetyl-CoA, except it's on an ACP. So we haven't done much different than we did in ketone body synthesis. And in fact, the next step is identical to it. We're going to take this beta-keto arrangement we still have, right, and reduce this carbonyl down to a secondary alcohol. We're going to use NADPH for this. In eukaryotes, it's done by NADH in prokaryotes, but not important for this class. But we're going to need a reducing molecule that gets oxidized in the process, right? It's almost always NADPH that we're going to use here in eukaryotes, and it becomes from a ketone to a secondary alcohol. So just like before, I have a 3, right, or beta hydroxy butyryl because it's got 4 carbons, ACP instead of CoA. But unlike ketone bodies here, we're not going to cut off the ACP, we're going to leave it attached. And we're kind of doing the opposite of what we did with fatty acid degradation. If you remember, our acronym for that was Ohio State. What's the opposite of Ohio State? And don't say Michigan. But what's the opposite of Ohio State? Let's read the letters backwards. So instead of doing oxidation, hydration, oxidation, sulfur transfer, let's reverse those processes. Right? I wish Michigan had this acronym, but it doesn't. It would be transfer off of sulfur, because the last step was transfer onto sulfur. So transfer off of sulfur, followed by reduction, the opposite of oxidation, followed by dehydration, the opposite of hydration, followed by reduction, again the opposite of oxidation. So following it backwards, we have transfer off of sulfur. Yes, I cut off the ACP. It's off of that sulfur. Then I do reduction. I get an alcohol. Dehydration, removing the alcohol, gives me my alkene. This is exactly the opposite of the other one. And then I reduce the alkene to an alkane using NADPH. Now you're thinking, hey, why didn't you use FADH2 here? Because it always uses NADPH for this pathway. Remember I said when we're doing oxidations, you can depend on that rule of NAD versus FAD. When you're doing reductions, it's almost always NADPH. So opposite of the sulfur transfer, opposite of oxidation, opposite of hydration, opposite of oxidation. And now I have 4 carbons put together. Now I want you to imagine taking this butyryl ACP at the bottom, butyryl because it's 4 carbons, and putting it right back at the top where this acetyl ACP was. So remove the blue one at the top, take the product at the bottom and put it right back on the top of the stack. Wouldn't I go through the same process again? A brand new malonyl ACP shows up, it decarboxylates, it attacks this beginning piece, which would be here at the bottom, right? I go through the reduction, dehydration, reduction again, and I would end up with a hexaryl ACP. It would now be 6 carbons long. Take that one, put it at the top, get a new malonyl ACP, and repeat and repeat and repeat, and I keep growing this fatty acid by 2 carbons every round. What does it cost me every round? Well, it costs me 2 NADPHs here to do every round. It also costs me making one of these malonyl CoAs every round, which is again an ATP. Right? So it costs me to do this, 2 NADPHs and 1 ATP to make the carbon chain 2 carbons longer. Right? And of course I'll keep making it until I get to the size I intend, 12, 16, 20 carbons, whatever it is. And then you've made fatty acids. Right? Do we want some double bonds put in them to make them unsaturated? Sure, that's a whole other set of reactions that we're not going to cover for this class. But what I want you to get out of this is you can see this fatty acid synthesis has 2 main parts. We're going to make the malonyl CoA as raw material, and we keep providing that as malonyl ACP to grow the chain by 2 carbons, doing the opposite of Ohio State. Okay. Finally, this isn't a new thing, right? The chemistry is new. It's a recent thing we've learned in the past few centuries. But ancient cultures also recognized that the liver was pretty important. Right? So they would, ancient Egyptians would remove organs, and they thought they had special things about them where the soul was contained, right? It wasn't in the heart, right? Or it was in the heart, not the brain. But they would take things out and store the liver and store the kidneys and store the other organs like the lungs. They thought these were important things. They didn't know why they were so important, but they had a knowledge of anatomy. And of course they had a knowledge of some medicine. And in the other cultures that came after that, like the Greeks and Romans, had myths that would explain things as well. And one of the myths was Prometheus, right? You've probably heard of it. I don't mean the movie. I mean the Greek god Prometheus. He was accused of giving fire to humans, right? It's something that the gods didn't want. That was their myth behind this. So Prometheus was punished by Zeus, and he was chained to a rock, and an eagle was to come along every day and eat most of his liver, right? Being a god, his liver could regenerate, right? And it would grow back overnight. The eagle would come around the next day and have the liver again. And that would happen every day while he was chained to this rock. A cartoon at the bottom showing that, one of the marble sculptures at the center left showing that as well. Right, so that was incorporated into their myth, but even then they knew that the liver could regenerate, right? If you destroyed 90% of it, as long as you leave the part around the hepatic portal vein, it would regenerate, right? So a lot of cultures knew this before we learned it in modern medicine.